Good evening, viewer. Welcome to our Glory Globe TV. Here's a breaking and most amazing news too. Yes, scientists at Tokyo Medical and Dental University have created a new genome editing methods that lowers the levels of proteins causing illness and associated problems in cells from a patient with Charcot-Marie tooth disease type 1A. In the past 100 years, scientific breakthroughs have greatly transformed our world. The field of genetics, in particular, has opened doors to a myriad possibilities, augmented human capabilities, cures for disease, and even changes to the course of evolution. In a study recently published in Communication Medicines, researchers from Tokyo Medical and Dental University have unveiled a groundbreaking genome editing technique. This innovation holds promise for treating Charcot-Marie tooth, that is called CMT, a relatively common hereditary nerve disease that affects the nerves and currently has no clinical treatment. Yeah, CMT is characterized by altered sensation and muscle weakness in the limbs and affects 10 to 80 people per 1 lakh. The most common CMT subtype is known as CMT1A and is caused by a duplication of the gene encoding peripheral myelin protein 22, that is called PMP22, leading to high levels of these proteins in affected individuals. Yeah, PMP22 is important for forming myelin structural unit, the fatty insulation that allows signals to rapidly travel from the limbs to the brain and neck. Also, the researchers have tried to reduce PMP22 in animal models of CMT1A using different techniques, but its translation to human patients has been unsuccessful. This may be because existing animal models do not have human-like PMP22 gene duplication. This study aimed to solve this problem. Yeah, innovative approach to treating CMT. We created a cell model by taking cells from a patient with CMT1A and growing them into Szechuan cells, which are the cells that makes myelin, says Dr. Hitoshi Okazawa, senior author of the study. We then used a specialized genome editing techniques known as AAV vectors to decrease the amount of PMP22 protein that was produced by the cell. Because both higher and lower PMP22 level can lead to different types of nerve disease known as neuropathies, the researchers had to be very careful about how much they reduced PMP22. They created and trailed different AAV vectors and eventually choose one that removed 20% to 40% of PMP22 gene copies from the genome. This was enough to reverse many CMT related changes in swan cell cultures and to improve the myelinating abilities of these cells, thus highlighting the potential of this treatment as a clinical therapy for the disease. Beyond the report, Glory Globe TV, Delhi, Bharat.